So thanks for that introduction. So today I'm going to talk to you about how we pick and place neuronal cells in the lab that I come from in LBB in order to create these neuronal networks. The focus in biology today is both on single cells and multi-cell organisms. And this is in particular the case when it comes to organ-on-chip devices, which might need more specific cell placement rather than just random distributions on the surfaces. So there's two approaches in neuroscience. There's a top-down approach whereby we look at the whole brain. This involves billions of neurons, and it is quite close to the real thing. However, it is extremely complex and therefore quite difficult to understand the intricate workings of it. So that's why in LBB, we focus on a bottom-up approach to neuroscience, whereby we just look at one unit of the brain, where we take as little neurons as possible in the order of 1 to 100 cells, and we try to create these neuronal circuits, which are a bit more reproducible for testing of our hypothesis. And then later we can build these up into more complex systems. So how do we build these neuronal circuits and test them? So we use these microelectrode arrays, which are essentially glass with electrodes at the bottom of them, and we can place the cells directly on top of them. And these electrodes can both stimulate the cells, and they can also record any electrical outputs from them. So on top of these microelectrode arrays, we then place these PDMS structures, and these are intended to guide the growth of the neurons in a particular way. So we have multiple examples of these configurations, so we don't always use this particular one, but I'll use this as an example to show you how we do it. So if we take one of these individual configurations, so the black area is a microchannel that is beneath the surface of the PDMS, and the white circles are the wells into which we seed the neurons. So this might be a little bit more clear if we take a cross-sectional view of it. So here we have the black section is the microchannel running beneath the surface of the MIA, and this connects the two wells into which we place the spheroids, or the cells. So we seed the cells into the wells, and then we leave them for a couple of days, and then they grow axons connecting the cells in each well between them. So the microchannels are just tall enough to allow the axons to grow beneath the surface, but it's not tall enough to allow the cell bodies to enter the channels. So you might have also noticed that these are shaped rather oddly, and this is because the shape provides directionality for the axons. So this encourages them to grow in a clockwise direction only, so we also have directionality of our cells. The blue dots that you might have noticed here on the four corners are where the electrodes of the MIA are placed beneath the PDMS structure. And we can provide an electrical stimulus at one of these, and we can read out the electrical activity of the cells from all of the others. And this gives us quite interesting raster plots here, where we can see quite often patterns in the firing activity of the cells when they're stimulated at a particular point. So I won't go into a huge amount of detail with the raster plots, but I'll explain to you how we then build our circuits. So as I was just saying, in an ideal world, when we seed our cells or our spheroids in on top of our PDMS structures, they fall nicely into the wells. And after a couple of days, the axons grow beneath the PDMS structure through the channels and connect to the cells in the next well. So unfortunately, we don't live in an ideal world. So when we seed the spheroids in on top of our PDMS structures, we get too many cells in some wells. In other wells, we don't get any. And then we also get cells seeded on top of our PDMS structures, which isn't ideal, as these can short circuit our other nicely grown circuits beneath. So in order to counteract this problem, we're using the FluidFM in order to directly pick and place our cells and spheroids into the PDMS structures so that we can get nice reproducible results with it. So I won't explain in too much detail how FluidFM works because I think you're all quite familiar with it. But in the application, we're primarily using the FluidFM OMNIUM. We're trying to make the whole pick-and-place process as automated and as quickly as possible. So to do this, for the single cells, we're using the flat probes with an aperture of 2 micron. And for the spheroids, we're using the larger apertures of 8 microns, but we're also transitioning now a little bit to use some of the tube tips that you might have seen in the presentations yesterday. So the pick-and-place procedure is fairly straightforward. The cantilever approaches the cell, applies a negative pressure, picks up the cell, transfers it to the new location where the cantilever is lowered, a positive pressure is applied, and the cell is deposited onto the surface. So because the FluidFM OMNIUM allows us to pre-program the points for picking and placing before we do our experiments, we can it's quite handy for our microstructures. We can pre-program these into it, and we can also create nice little images with the cells. So this is meant to be the lab logo. So if we look at how it's doing, the cell transfer time is quite fast. It's about 35 seconds. We have a nice placing success rate of about 83%. However, the picking success rate is at around 52% at the moment. So one of the reasons why it's a little bit low is because if we select a large number of cells, say 30 at a time, by the time the OMNIUM has reached cell 7 or 8, the cell might have moved a little bit, so the cantilever is just picking empty air. So another problem that we had, sorry, I forgot to mention earlier that the cells that we are using are primary rat cells, but we're also now transitioning to iPS-derived cells. So iPS-derived cells are great in that they more replicate human cells. However, one of the problems that we have with them is we don't derive them in-house. We actually buy them in frozen and thaw them out. So when we do this, unfortunately, we have quite large problems with viability. So we're hoping also to use the FluidFM in order to help rectify this by just picking, let's say, the good cells. So we can do this by taking our cells and staining them with our live dead assay, and using the fluorescent capabilities, we can then manually select our live cells and pick them and place them into our structures. However, we would prefer not to do this because it has been shown that the live dead stains have a negative impact on the cell viability over time. So what we would like to do, and what will hopefully also counteract the problem that we're having with the cell movement, is if we can get a cell recognition with the ARIA, we can, using the bright field images, select the good cells, and this will allow the ARIA to automatically select the cells that we want, which will make it a lot easier to pick them. So in order to do this, the ARIA software can capture an image of the screen, export this to an external script, which then processes, detects the points of the cells, and returns these points to the ARIA. So we have 2 methods of doing this right now. So I'm working on this with 2 PhD students in our lab, Katerina and Joelle, in order to do this. So currently we're working on 2 methods in order to figure out which one is the best. So the first we have is image segmentation, where the images are divided down into much smaller ones, and the cells are detected and then provided with a score based on their size and shape. And so this helps us to eliminate any cell aggregates that might show up, which we don't want to pick, or it can also eliminate any smaller cells, which might actually not be cells, but rather cell debris or something like this. The other method that we're using is cycle GANs. So this has previously been used in LBB for a different application, but we're hoping to transfer it over. It's more of an image or a machine learning technique whereby we create these images with the fluorescent images that I was showing you earlier. We overlay the live dead results onto the bright field images, and we can use these to train the algorithm. So by inputting then our bright field image, the algorithm creates this new image with the live dead cells outlined, and then it can return these points to ARIA. So how is it working? So the image segmentation, the advantages are it's quite fast relative to the cycle GANs approach. It only takes a couple of seconds. However, the problem with this is that it can't yet distinguish the live dead cells. With the cycle GANs, it can work reasonably well to identify the live cells. It can distinguish them with about 85% accuracy, which is really cool. It also works well with terrible backgrounds, which can occur quite frequently sometimes if your glass isn't clean enough. But one of the disadvantages of the cycle GANs is it's quite slow, and it cannot distinguish the dead cells so well yet. So we're still working on this to try and improve it all of the time. So hopefully in a couple of weeks' time, it'll be working even better. So currently we're primarily using the image segmentation approach. It takes about 15 seconds to run with ARIA. And we can use this both at the beginning of the experiment to help us pick out the locations of the cells we want, but also during the experiment in order to ensure that the cell hasn't moved around a little bit during the previous picking and placing experiments. So that's the cells. The other thing that we deal with are the spheroids. So these are also created from either the iPS-derived neurons that I was talking about earlier, but also from primary rat cells that we get. So the method is quite similar to the method for the pick and place of the cells, just slightly different parameters, whereby we pick with a negative pressure, relocate, and place it again with our positive pressure. So again, we were able to have quite a number of applications using the OMNIUM. So in one case, we like to grow our neurons quite often with astrocytes, which are kind of a helper cell, and help them to survive quite nicely. So in this, we unfortunately cannot control the amount of neurons that we place into each spheroid. So therefore, we statistically see them so that we have one neuron in each spheroid, and then we stain our neurons. So using the fluorescent capabilities of the OMNIUM, we can then pick out the spheroids that have 1, 2, or 3 neurons, however many we want, and pick and place these into our structures. So this is just an example of one that was picked in place using this technique and grew up after a few days. There is a little bit of undergrowth, unfortunately, under the structure. You can see it's not quite in the stomach shape as this side, but it grew quite nicely following the pick and place. Another thing we can do is a little bit like the cells that you saw earlier. We can also pick and place into nice formations on flat surfaces. Because the spheroids are a little bit wobbly on the end of the cantilever, maybe it's not quite as accurate on the flat surfaces with picking and placing as it is with the cells. However, I still like to think that it's a somewhat recognizable LBB. As you also saw in some of the videos earlier, we can also place directly into the PDMS structures. This is a little bit more accurate because you have the nice guidance of the walls to guide the spheroid in a little bit more easily. And we can also pre-program the points of the wells into ARIA, so it makes it nice and fast. So in this example, it takes about 60 to 90 minutes to do this. And finally, we can also place the spheroids directly onto the multi-electrode arrays. So this is an example here. The black dots are the electrodes with the PDMS structure mounted on top of that. So yeah, we can place our spheroids directly in there. So with the pick and place of the spheroids, it's a little bit slower than the cells. Our placing success rate is nice as well. We've 97% success. Our picking rate is also not as great, but it's something that we're actively working on at the moment to improve that. So to conclude, the cells and spheroids can be placed in defined formations on flat surfaces. They can be selected based on the cell type, and also we're currently working on selecting based on viability. And finally, the spheroids can be placed within defined formations within a PDMS structure and also on the multi-electrode arrays. So thank you very much for listening. I'd like to thank my group in LBB, Cytosurge, and InnoSuisse, the funding body. And thank you for listening.